Pseudohabdosynochus. Pseudohabdosynochus is a genus of monopisthocotylean monogeneans, included in the family Diplectanidae. The type species of the genus is Pseudohabdosynochus epinepheli Yamaguti, 1938. The genus includes more than 80 species, which are all parasitic on the gills of marine fish, especially groupers. Since groupers are mostly abundant in warm waters, species of Pseudohabdosynochus are found in warm parts of the oceans, especially in coral reefs, but species have also been found in deep-sea groupers. Several species of Pseudohabdosynochus are pathogens of maricultured groupers. Morphology. All species of Pseudohabdosynochus are small animals, ranging 0.31 mm in length. As most monogeneans, they are flat, with an anterior head bearing 4 oculi and head glands, a main elongate body, and a posterior hapter. The digestive system includes an anterior muscular pharynx and 2 lateral intestinal branches or ceca. As in all platyhelminths, there is no anus. The hapter, in the posterior part of the body, is a specialized organ used to attach to the host. The hapter includes sclerotized elements, namely a ventral bar, 2 lateral dorsal bars, 2 ventral hooks and 2 dorsal hooks, and 14 hooklets. As in most diplectanids, the hapter bears special, characteristic structures called squamodiscs. The squamodiscs, 1 ventral and 1 dorsal of species of Pseudohabdosynochus, are made up of numerous rodlets aligned as concentric rows. All species of the genus have 2 squamodiscs except Pseudohabdosynochus syndiscus nefari and use it 2007, in which these organs are completely lacking. Adults are hermaphroditic. The reproductive organ include a single ovary and a single testes. As in all diplectanids, the ovary or germarium is anterior to the testes and loops around the right intestinal cecum. Species of Pseudohabdosynochus are characterized by a sclerotized male copulatory organ or quadriloculate organ, which has the shape of a bean with 4 internal chambers. The vagina includes a sclerotized part, which is a complex structure with several chambers and canals, generally used for the diagnosis and differentiation of species. Etymology and nomenclatural case. The origin of the name Pseudohabdosynochus merits an explanation. Pseudo from Greek pseuds lying. False is a prefix commonly used in creating a new scientific name for a taxon that superficially appears to be a taxon, but actually is another. The famous Japanese parasitologist Satoru Yamaguti described a membranous plaque on the posterior region of Pseudohabdosynochus epinepheli and created the generic name Pseudohabdosynochus in reference. In his interpretation, also had lateral plaques. It has been shown later that plaques in P. epinepheli was an erroneous interpretation of damaged specimens. Moreover, the diagnosis of Rhabdosynochus was later modified, and it is now considered that species of this genus do not have plaques. Therefore, the genus name Pseudohabdosynochus is the result of both an erroneous observation and an erroneous comparison by Satoru Yamaguti. However, it is valid according to the Ixon. Pseudohabdosynochus is also an interesting nomenclatural case. Yamaguti described the same species twice, the first time in 1938 as Diplectanum epinepheli Yamaguti, 1938, and the second time in 1958 as Pseudohabdosynochus epinepheli Yamaguti, 1958. In both papers, the species was described from parasites collected from the same fish species, the grouper Epinephelis acara off Japan. Kritsky and Beverly Burton, 1986. Solved this case with reference to the International Code of Zoological Nomenclature. The type species of the genus is the name Pseudohabdosynochus epinepheli Yamaguti, 1958. Pseudohabdosynochus epinepheli Yamaguti, 1958, is a junior synonym of Pseudohabdosynochus epinepheli Yamaguti, 1938. Kritsky and Beverly Burton, 1986, and Pseudohabdosynochus epinepheli Yamaguti, 1938. Kritsky and Beverly Burton, 1986, is the modern valid combination. The latter species, a taxon, and the type species of the genus, a name, are considered to correspond to the same biological taxon but have different, although valid, names. Recent re-examination of type specimens have confirmed this interpretation. Hosts. Species of Pseudohabdosynochus are parasitic exclusively on the gill filaments of marine fish. Most of these fish belong to the family Serranidae and are commonly known as groupers, such as many species of the genus Epinephelis, and also species of Cephalophalis, Nycteroperca, and Variola. A few species have also been described from fish of the genera Alphestes, Paranthias, and Paralebrax. As most Diplectanids, species of Pseudohabdosynochus are generally strictly species-specific, i.e. a species is found on a single species of host and no others. However, several exceptions have been reported. Some species of groupers harbor a wide biodiversity of Pseudohabdosynochus species and sometimes other monogeneans as well. Examples are Epinephelis maculatus, which has 8 distinct Pseudohabdosynochus species, and Epinephelis malabaricus, which has 7 species. Life cycle. Cycle. The life cycle of Pseudohabdosynochus species is similar to that of other diplectanid monogeneans. It is monoxenous, i.e. there is a single host, the fish, the fish, the fish, the fish. The life cycle includes a parasitic phase, the adult worm, and 2 stages, which are free in the environment, the egg and the larva. The adult hermaphroditic worm, which lives on the gills of fish, lays eggs in the water. The egg has an operculum. A larva develops in the egg and hatches through the operculum. The swimming larva is ciliated and called an onchimericidum. The larva finds its host by swimming to it, first attaches to the skin and migrates to the gills, and eventually transforms itself into a young monogenean, which is attached to the gill filament by its haptor. During transformation from larva to adult, the monogenean loses its cilia and produces a new tegument, a phenomenon which is considered a general characteristic of the parasitic platyhelminths or Neodermata. Information is available for the life cycle of Pseudohabdosynochus lantoensis, a parasite of the orange-spotted grouper Epinephelis coides, at a temperature of 30 deg C. The adult lays 10-22 eggs/day. The eggs hatch within 2 to 4 days. The onchimericidum is viable for -8H and should find a host within this delay. After attachment, the onchimericidum transforms into an adult in 4-7 days. The parasite produces eggs after 7 days. The life cycle is thus complete in 13-20 days at 30 deg C. Certain species of Pseudohabdosynochus parasitize groupers which show spawning aggregations, i.e. the adult fish gather annually at a precise location and meet other fish of their own species only once per year. It has been suggested that transmission of Pseudohabdosynochus species occurs preferentially during these spawning aggregations, from older infected fish to younger uninfected fish. Pathology and control in mariculture. Species of Pseudohabdosynochus are attached to the gills of groupers. No information is available on the pathogen effect of these parasites in wild groupers. Since the life cycle is monoxenous and thus can be completed with a high rate of success in mariculture condition, species of Pseudohabdosynochus can proliferate on gills of cultured groupers and become dangerous for the fish, inducing mortality. Parasite infections are often measured by 2 variables: prevalence, the proportion of infected hosts, and intensity, the number of parasites per host. Prevalence is as high as 100%, i.e. all fish are infected have been reported for Pseudohabdosynochus sp. In cage-cultured Epinephelis malabaricus in Thailand for Pseudohabdosynochus sp. On Epinephelis fuscogutensis and Epinephelis coides maricultured in Indonesia, and for P. beverlibertani on Epinephelis marginatus, wild or maricultured, in Brazil. Intensities are as high as up to 1006 individual Pseudohabdosynochus sp. per fish Epinephelis fuscogutensis in Indonesia, up to 294 p. coideces per fish Epinephelis coides in South China, 327 p. eucatensis per fish on Epinephelus morio in Mexico, and 500 p. epinepheli per 100 grams of gill filaments of Epinephelus acora in Japan. Eradication of gill parasitic monogeneans is a problem because various chemical treatments can be harmful for the fish. Treatments used against Pseudohabdosynochus species include freshwater, diluted formalin, and diluted hydrogen peroxide H2O. Diluted hydrogen peroxide H2O seems the preferred treatment for P. epinepheli on Epinephelus acora and P. lantonensis on Epinephelus coides. List of species. Lists of species have been provided by Kritsky and Beverly Burton 1986 and Justine 2007. The list below, about 80 species, is based on the World Register of Marine Organisms with additions.